Hello guys, so welcome to our new video of our channel Textbook Reading. So today this is the video of continuation of part 1 and part 2 of chapter 1, Cell Structure and Functions. So let's get our biology textbook. And here is our biology textbook. So let's open to chapter 1. In part 1, we read page number 1 and page number 2. In part 2, we read page number 3 and page number 4. Now, let's read page number 5, that is the continuation part. This is very similar to cell membrane. Almost the entire genetic material of the cells is found in the nucleus. On the basis of the presence or absence of the organized nucleus, cells are categorized into two types, that is prokaryotic cell without organized nucleus and eukaryotic cell with organized nucleus. Prokaryotic cells, the above description was primarily about eukaryotic cells that contain a membrane-bound nucleus. Cells that do not have nuclear membrane-bound nuclear material are called prokaryotic cells. We have mentioned earlier that the bacterium is prokaryotic cell. Cyanobacteria, blue-green algae also belong to this category. Cytoplasm. When we look at the temporary mounds of onion peel, we can see a large region of each cell enclosed by cell membrane. The region takes up very little stain. It is called cytoplasm. The cytoplasm is fluid content bounded by the plasma membrane. It also contains many specialized organelles. Each of these organelles performs specific function for the cell. The cell organelles are enclosed by the membranes. In prokaryotes, besides the absence of defined membrane-bound nucleus or nuclear region, the membrane-bound organelles are also absent, except membraneless ribosomes. Protoplasm versus cytoplasm. For a long time, it was believed that the essence of life was stored in the fluid found inside the cell. Hence, this was named protoplasm, which means life fluid. But when it became clear that the fluid is basically a medium in which various particles and membranes float around, the functions of the cell actually take place in these organelles, it began to understood that life resided in these organizations. In particular, the material inside and outside the nuclear membrane was differentiated after discovery of the nucleus. Hence, protoplasm was renamed as cytoplasm, that is cell fluid. The fluid inside the nucleus came to be known as the nuclear fluid or nucleoplasm. Cell organelles. Now let us discuss some important cell organelles. Mitochondria, plastids, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. They are important because they carry out very crucial functions in the cell. Endoplasmic reticulum. When the cell was observed under the electron microscope, a network of membranes was observed throughout the cytoplasm. This network creates passages within the cytoplasm for the transport of substances from one part of the cell to another. This network of membrane is known as endoplasmic reticulum. The endoplasmic reticulum (ER) is a large network of membrane-bound tubes and sheets. The ER membrane is similar in structure to the plasma membrane. Endoplasmic reticulum may have some granule-like structure on its surface, which are called ribosomes. Such ER is also called rough endoplasmic reticulum (RER). Area sections of the RER that do not have ribosomes on them are called smooth endoplasmic reticular SER. Rough endoplasmic reticulum is the site of protein manufacture. The SR helps in the manufacture of fat molecules or lipids important for cell function. The manufactured proteins and lipids that are sent to various places in the cell depending on need using the ER. Some of these proteins, and lipids help in building the cell membrane. Thus, one function of ER is to serve as channels for transport of materials, especially proteins, between various regions of cytoplasm or between the cytoplasm and nucleus. It also functions as cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. In vertebrate liver cells, SER plays a crucial role in detoxifying many poisons and drugs. Golgi body or Golgi complex. Although Camillo Golgi had observed this organelle in year 1898 using a compound microscope, its finer structure could be observed only under electron microscope. This organelle is made up of several membranes. These membranes create sac-like structures around them, which many fluid vesicles abound. The proteins and other substances produced in ribosomes reach the Golgi body through these vesicles. Here, these substances are altered slightly. In one sense, the function of Golgi body is to pack various substances before they are transported to other parts of cell. From here, these substances are either sent towards the cell membrane or to another organelle, the lysosome. After reaching the cell membrane, these substances are secreted from the cell and sometimes even used to regenerate or repeat the membrane. The number of Golgi bodies varies from cell to cell. Their number is particularly large in those cells that secrete hormones and enzymes. So guys, as I said, this is Biology Class 9, Chapter 1, Cell Structure and Functions, Part 3. Watch Part 4 for the entire lesson. So this is the end of the video. Don't forget to like, share, subscribe, and don't forget to hit the bell icon. Thank you for watching.